You're listening to Conferences on Lyme Allergy from Children's Mercy Hospitals and Clinics in Kansas City, Missouri. Today is May 14, 2012, and I'm your host, Dr. Jay Portnoy. Our topic today, Immunomodulatory Therapy. Our presenter is Dr. Lanny Roswasser. He's a professor of medicine and the D. Lyons, Missouri Endowed Chair in Immunology Research at Children's Mercy Hospitals and Clinics. Well, without any further delay, we'll go ahead and introduce Dr. Lanny Roswasser. Dr. Rosenwasser is the D. Lyons, Missouri Endowed Chair in Immunology Research here at Children's Mercy Hospitals and Clinics. He's also a professor of medicine at the University of Missouri Kansas City School of Medicine. He's an internationally recognized expert in the field of immunology, and he's agreed to speak with us today about immunomodulatory therapy. So without any further delay, I will turn the podium over to Dr. Lanny Roswasser.
Thanks very much, Jay.
Here's your mouse.
Great keyboard. It's got everything I need for this. It looks like, whoa. And that's the first slide, which is my disclosure, always the beginning of it. So I haven't had 100 shares of anything lately, and this is my disclosure for the past year or so in terms of research studies, consultancies, and speakers bureau. So today we're going to talk about immunomodulator therapy and biotherapeutics. I've given a call on biotherapeutics a few months ago. This will update some other information that we've had in that previous presentation. I don't know if it's in podcast, but you can compare the two if you'd like. This particular talk is companion to the updated primer that was in the JACI a year or so ago. And there's a chapter on immunomodulators written by Artie Cavanaugh and his colleagues in San Diego. And so this is sort of a companion piece for that, chapter. Most of the things that he has listed there we'll touch on, and then there's some different approaches and some updates that we'll have that are a little bit different than what Artie and his colleagues have written. So the learning objectives are listed here, concept of biotherapeutics, as opposed to the Cavanaugh review. We're going to concentrate a little bit more on allergic responses and asthma in terms of immunomodulatory effects. There's some very interesting current preliminary studies, potential biotherapeutics, and the three areas that we'll touch on with this are inhibition of IL-1 and the expanded knowledge we have of the IL-1 family of modulators and potential regulators, some newer data with anti-IL-13, and some other information with anti-IL-17, not so much for allergic disease and asthma, but for psoriasis. But the application of that data related to psoriasis has big implications for Th17 cell neutralization, which also potentially could have some very interesting effects in allergic responses and asthma, as we'll discuss. So there are many complex cascades in immune responses in general and in allergic responses that play a role here. Biotherapeutics is a field that encompasses materials, usually proteins, produced by biologic means, including recombinant DNA technology. Agents, antagonists, agonists for treatment are usually biological, and some are naturally biologic, some are synthetic. The agents that are used include monoclonal antibodies that can be directed to cell surface receptors, to ligands, to microorganisms. Cytokines and soluble receptors, we'll mention that. There are natural and synthetic antagonists that one can use that are either protein or small molecule. And the small molecules may be designed or modeled based on mechanisms, such as the small molecules that inhibit kinase activity with cellular activation. And there are small inhibitory RNAs that will inhibit the transcription of particular messenger RNA and expression of targeted genes at the siRNA, generally knocked out by families based on motif recognition of the RNA. Oligonucleotides about 10 years ago were of interest, mainly because some of them, such as CpG, seem to inhibit immune deviation and responses skewed towards allergic as opposed to host defense responses. Some of that hasn't been as profitable in terms of its ability to really influence the outcome of allergic responses or asthma. And there are antisense oligonucleotides that in a sense are synthetic equivalents of the natural siRNA molecules that are used to regulate. And then there are other kinds of inhibitors, some of which we'll touch on, and they're not in the Kavanaugh chapter, and they're listed on a couple of slides in my presentation. I think the presentation will be archived so it can be downloaded. Yes. So even though there's no CME, you can look at some of these responses, or if you're interested in looking at some of them, it'll be available. So in terms of biotherapeutic responses, anti-IgE for allergy and asthma is the only one that we really have that's approved, but there are huge numbers of innate immune targets, either related to the pro-inflammatory cytokines associated with innate responses like IL-1, TNF, or IL-6, toll-like receptor adhesion molecules, interferon modulation, chemokine receptors, all are huge numbers of potential targets. Then there are responses related to acquired immunity, Th2 and Th17 cells in particular, the cytokines they make, and the cells in particular, monocytes, macrophages, dendritic cells, and T and B cells involved in acquired immunity, multiple targets. And then there are other things, growth and differentiation factors. No question that material like TGF-beta is critical in the lung and in the skin and in the GI tract in terms of physiology and regulation. Adipokines is one of the ways in which asthma and allergic responses may coincide with some of the dysregulation found in obesity. And then the epithelial protein known as TSLP, thymic stromal lymphopoietin, excuse me. TSLP is an important epithelial product that will skew responses towards an allergic response. In terms of asthma, the definition requires 4 components in terms of asthma. But the thing I wanted to show is the fact that we're looking at multiple cascades in asthma that interact. The complexity just of the Th2 responses are seen in this end of the picture, and the complexity in terms of the growth and differentiation factors, including TGF-beta and other fibroblast activation, growth factors, smooth muscle active factors, all are involved in the generation of a hyper-responsive airway structure in asthma as the model. That there are clearly skin changes in terms of epithelium and subepithelium one finds in allergic dermatologic conditions, and in the GI tract and food allergy and other allergic conditions like eosinophilic esophagitis. Some of the changes in the actual structure of the target organ are important. As I'll show you here, these responses now are being dissected at a very more precise level. This is from a publication in the New England Journal, an editorial that accompanied the work on anti-IL-17 or lebrikizumab, or excuse me, anti-IL-13 or lebrikizumab, showing an effect in asthma, which we'll talk about some of the data in a moment. But there is the identification of the production of periostin by the epithelium under the influence of IL-13 generated by Th2 type immune reactions as being a prototypical marker of the epithelial response to this asthmatic milieu. And the periostin can be detected not just locally in bronchoalveolar lavage, but it's also possible to detect it in the serum. And elevations in periostin in the serum may be a marker in the subset of asthmatics who respond to anti-IL-13 and who presumably have this Th2 IL-13 dominant immune response in their airway or in the airway subepithelium. Of interest is it's been worked out by some of the cell biologists at UCSF that the actual secretion of periostin under the influence of IL-13 by the epithelial cells is actually basal rather than at the edge of the epithelium. So there is presumably more periostin being generated heading towards the circulation as opposed to the airway. That's one of the reasons why a serum measurement of periostin is presumably a little bit easier to correlate with this Th2 type response in the lung. So this has garnered a lot of attention in the last 6 months or so as a way to sort of differentiate those who might have a differential response to some of these immunomodulators, and we'll look at some of that data in a second. Acquired immunity, not just innate immunity, plays a significant role, especially in asthma, where we know where the prototype of allergen and or viral protein activating predominantly Th2 cells in airway and in other allergic tissues leading to allergic diseases is a model. The differentiation of these T cells into different diverse subsets is very important. The concept of there being a Th1, Th2 balance, so predominant in the thinking concerning pathogenesis of allergic disease a decade ago, now recognizes the general diversity of the CD4 acquired immune response, the role of T regulatory cells that control the activity of all of these types of cells on an antigen-specific or antigen-specific manner, the ability of the T regulatory cells to generate TGF-beta and IL-10 both at the surface of the cell and via secretion is critical to the inhibition of some of these other cells. The T regulatory cells not only inhibit the Th2 cells, but they also can inhibit Th1 and also Th17 cells. So these are all potential targets in asthma, allergic diseases, allergic skin diseases, and food allergy. Clearly the ability of T regulatory cells to inhibit Th2 is critical. The role of increasing the T regulatory cells during allergen-specific immunotherapy is well established. It's one of the accepted mechanisms of many of the different kinds of mechanisms for immunotherapy working. These cells undergo differentiation based on the milieu in which they are stimulated. So if there's a milieu where there's a large amount of IL-4, the naive cells will transcribe GATA3 and STAT6, which has an influence on the downstream production of the cytokines and other molecules involved in allergic responses and allergic regulation. Th17 cells depend on IL-6 critically, along with TGF-beta, to induce STAT3 and ROR2C, which is the human equivalent of the mouse transcription factor ROR gamma T. So this is updated now, would be ROR2C in humans, to develop Th17 cells. In the skin, the Th17 cells further differentiate into the cells that produce IL-22, a prominent part of atopic dermatitis, but it can also be seen in psoriasis. So there's a subspur of differentiating cells. These cells were once thought to be terminally differentiated into these different phenotypes, but it's now clear that there's plasticity in this response. So there are some Th1 cells never terminally differentiate, but that can go back to a naive phenotype and be stimulated in different circumstances as the milieu changes. These are all important because all of these factors then become ways in which you can modulate an allergic response. And as you'll see, some of these things are getting to the point where they can potentially be utilized. The T regulatory cells express TGF-beta and IL-10 on the surface membrane, but they also secrete those cytokines. And it's found now that both kinds of mechanisms are involved in suppression of these other T cells. TGF-beta's role is specific at inducing FOXP3. CD4 positive lymphocytes, when they get activated, could express CD25. But if there's no TGF-beta in the milieu where that happens, you just have an activated T cell. When there's TGF-beta in the milieu, you get FOXP3 activated, and you can generate a true T regulatory cells that can inhibit the other T cells. IL-35 is a unique growth factor for the T regulatory cells. These allergic responses are highly complex. There's sheets of these cascades that have to occur in some kind of order with some kind of interaction to generate a true disease state of allergic disease, asthma, atopic dermatitis. So showing these cartoons and talking about them is one thing, but it's actually highly complex. Unlike our schemes to actually treat asthma, as you can see, this is a very simple and straightforward approach to the treatment of asthma. For those of you who can't see me, that's tongue-in-cheek. So the complexity of our treatment schemes, I think, reflects our relative lack of full understanding of the whole complexity of the pathogenesis. So we have 6 steps, and some of the steps require certain kinds of treatments, and other steps require other things. You have to assess risk, control, and then slide your patient across this wide range. And it's hard for specialists to keep up on every bit of data related to this kind of thing. I really am. I have a lot of sympathy for primary care doctors who try to understand what this might mean in terms of caring for asthma patients, because it is very complex. The only biotherapeutic and immunomodulatory material we have in allergy and allergic disease, in particular asthma, is Omalizumab, which is anti-IgE, and it's been shown to have efficacy in a variety of circumstances, but at the present time it's only approved for asthma. In the beginning, about 12, 15 years ago, when these mechanisms were being uncovered, a variety of tests of different kinds of biotherapeutics were designed. Some of them are listed here. I've been showing that for a long time. This comes from a review that Paul Verne wrote that chronicled the failure of about the first 8 or 9 of the biotherapeutic interventions in asthma, starting with the very limited treatments with anti-IL-4 and anti-IL-5 for asthma that were had some points where they looked promising, but then as they were expanded and looked at in different ways, they were less than efficacious. Even TNF was also a reasonable site for blockade. The great efficacy of TNF blockade with either the receptor antagonists or with antibodies to TNF in the rheumatic diseases in a variety of circumstances, especially in rheumatoid arthritis, made that a very attractive thing to look at. The group in Leicester in the UK looked at a trial over 10 weeks of etanercept, which is the Enbrel, the TNF receptor antagonist that blocks TNF, and there was a suggestion that there was an effect on post-bronchodilator FEV1 over the 10 weeks in this very small trial of maybe 6 or 10 patients. Led to a larger trial of about 300 patients. The incidence of 7 diverse types of tumors in that trial of 300 or so patients made TNF blockade and its use in asthma and presumably other allergic diseases something that was unacceptable as a potential risk, even though the linkage to why it occurred is not well defined. It's an accepted risk related to TNF blockade, and even though there was some glimmer that TNF blockade might work well in, as a biotherapeutic treatment for asthma, it's been dropped. I have an interest in IL-1. There's a large group of genes on chromosome 2q13 that code for IL-1-like proteins. The prototypes are IL-1 alpha and beta and IL-1RA. IL-1RA is a natural receptor antagonist for IL-1, and if tenfold greater amounts of IL-1RA is made, it will inhibit the ability of IL-1 alpha or beta to bind to its various receptors. And this IL-1RA became Kineret, which is an anti-arthritis agent. So blocking IL-1 as part of innate immunity may also have a similar effect to blocking TNF. But unlike TNF, which is the receptor is tied in to death sequences and the possibility of tumors, IL-1 blockade does not seem to have that kind of risk associated with it. Kineret is also called anakinra, and it's already approved for a variety of indications: juvenile arthritis, regular rheumatoid arthritis, and it's being tried in a variety of other kinds of circumstances where it's been shown to have efficacy, which we'll mention. IL-18 is in the IL-1 family. It's a pro-inflammatory agonist, and it activates both acquired and innate immunity and Th2 responses. So IL-18, even though it can activate interferon production, also can activate Th2 arms of immunity. IL-5, 6, 7, 8, 9 are making up what's known as IL-36, IL-37, and IL-38. I'll mention those in a second. IL-33, which is IL-1F11, binds to Th2 cells through the ST1 receptor, and it's an important potential activator of Th2 immunity. It's also an activator of innate immunity. So many of these proteins activate dendritic cells and epithelium and other things that one would normally associate with innate immunity and host defense. There are natural receptor antagonists similar to IL-1RA, such as IL-37 and this IL-1HY2, which I think is going to be IL-36 alpha or something. I've got it in the next one. There it is. IL-1F5 is IL-36RA, so it's a receptor antagonist. F6, 8, and 9 are alternatively spliced forms of the same gene. So there's IL-36 alpha, beta, and gamma. I didn't write them down. IL-F7 and IL-37, which were already mentioned, which is an IL-1 receptor antagonist. And IL-1F10 is going to be IL-38. So most of the IL-30s, when you get a test on them, various places are all part of the IL-1 family. And there's going to be some potential for some new treatments that will come out of that. Just to go back, what's been found that recently, both in humans and in mice, is that IL-1 is a critical cofactor for both Th2 and Th17 T cell activation. In vitro and in vivo, Bill Paul has done the mouse work related to this, and he's very excited about this as a potential use of IL-1 as an adjuvant for vaccine. But it's also possible that blocking IL-1 may help with blocking Th2, Th17 T cell activation. So that becomes another reason to think IL-1 blockade in the treatment of asthma might help. We've done a lot of work related to IL-1 T cell activation, which Bill Paul doesn't, when he spoke in Aspen last year, didn't quite recognize. But I had to remind him that we cloned IL-1 based on its T cell responses. And there's no question that IL-1 is upregulated in asthma and other allergic diseases at the site of the late phase and also directly in the lung. So we've already talked about this IL-1 family. There actually is an approved treatment of a number of diseases, most of which relate to cryopyrin-associated periodic syndromes, or CAPS, that Hal Hoffman has written and spoken about. Familial cold urticaria is the prototype for this, but Muckle-Wells and the Nomad syndrome all respond to anti-IL-1 treatment utilizing Kineret or anakinra. There's also targeted therapy with IL-1 in gout, pseudo-gout, type 2 diabetes, and other kinds of inflammatory circumstances. And there are reports that are non-randomized, non-placebo-controlled case reports related to efficacy of IL-1 inhibition in Still's and adult-onset Still's and Schnitzler's. So there's a variety of disease categories where it's worked. That would make you think that neutrophilic allergic disorders might also be helped by IL-1 inhibition. There are 3 drugs now approved in these CAPS syndromes for use. Anakinra, IL-1RA, has been approved for a number of years for rheumatoid arthritis, so that's use has been involved in that treatment. The problem with IL-1RA, as I mentioned, you have to get significant concentrations of the IL-1RA to be sure that all of the IL-1 alpha, beta, and some of those other products that are agonists are completely blocked. So the treatment with anakinra or Kineret is a daily injection of the IL-1RA, and it's a pretty high concentration. So there's a lot of potential for local pain, and in kids, that's, I think, a needle a day or a shot a day of a real significant material is different. I have a number of patients with adult onset CILs. Some of the fellows have seen them over at Truman who've done remarkably well with Kineret or anakinra as the treatment. So you have to write a lot of letters to get their insurance to cover it because it's an expensive treatment, but it's very efficacious in these unusual syndromes. Canakinumab has been developed. It's also known as Alaris, that blocks IL-1 alpha and beta directly on cross-reactive epitopes, and also is efficacious. And since it's an antibody, an injection every 2 weeks or every 4 weeks, or an infusion is its preferred treatment, so it's got an advantage over Kineret. Rilonacept is a soluble inhibitor, much the same way that etanercept is a soluble inhibitor for the TNF molecule. Rilonacept is actually two IL-1 receptors that are hooked together along with an FC receptor. So it's also known as an IL-1 trap because it's not the natural receptor. It's actually designed specifically for inhibiting IL-1 and has been shown to be efficacious. This would require injections about once a week. So it's got a little bit of a different profile. This is just what the rilonacept looks like. It was approved for the CAPS, and a couple hundred patients receive it. Canakinumab is Alaris. It's a fully humanized anti-IL-1 alpha. It also blocks IL-1 beta. Or, excuse me, it's an anti-IL-1 beta that blocks IL-1 alpha binding as well, so it blocks both. IL-17 has become a very critical part of the inflammation associated with a number of immune-based diseases, but in particular even asthma and other allergic diseases utilize Th17 cells as effector immunocytes, even though they don't make the classic allergic cytokines. They make IL-17. There's actually a family of cytokines here that are made up of 6 cytokines. IL-17A and F are profibrotic cytokines that can activate chemokines and also generate an amplification loop that activates IL-6. IL-17E, the 5th one, is also known as IL-25, and that's an activator of Th2 cells, and obviously it's associated with eosinophilia and airways hyperresponsiveness. Genetics of a number of genes within this IL-17 family have been linked to asthma as well, so there's no question that IL-17 and Th17 plays a role. About a month or two ago, there was back-to-back papers in the New England Journal that identified monoclonal antibodies to targets in the Th17 pathway that had efficacy in psoriasis. So there's one called ixekizumab that targets IL-17 itself directly. There's one brodalumab that blocks the IL-17 receptor, and there's one called tocilizumab that blocks the IL-6 receptor, and this prevents Th17 cells from being differentiated into effector cells. So this one actually has also been not just evaluated in psoriasis, but trials in Japan have looked at it in rheumatoid arthritis and SLE. But these all potentially will get targets, I think, that are active in asthma and atopic dermatitis. So they can, I think, be applied to allergic diseases as well. So there are a number of factors to inhibit Th17 cells that are on the horizon. There have been a number of studies in the past 2 or 3 years that demonstrate that anti-IL-5 actually does have an effect in asthma, as well as in hypereosinophilic syndrome, an unusual syndrome that we've discussed in other venues. But it's steroid sparing in both of those syndromes. with both mepolizumab and resolizumab, which are both anti-IL-5 antibodies. None of these have been approved, even though they've been shown to have efficacy in both hypereosinophilic syndrome and in asthma in terms of steroid-sparing activity. There's another factor known as MEDI563, which blocks the IL-4 receptor and has a similar profile and effect that the mepolizumab and resolizumab have. This just demonstrates in two different studies that the mepolizumab inhibits exacerbations of asthma in steroid-dependent asthma where sputum eosinophilia is detected. The group at Leicester, UK, Haldar and Wardlaw and others, and then Param Nair at McMaster in Canada published this back-to-back in the New England Journal a couple of years ago. And not only does it drop the eosinophils, it improves FEV1 as well as quality of life and PC20 in terms of bronchial responsiveness. Lebrikizumab is anti-IL-13. There was a study recently done by Genentech and Roche, which looked at moderately severe steroid-dependent asthma, in which lebrikizumab was given for 6 months with an 8-week follow-up period, a 2-week run-in, and randomization with either lebrikizumab or a placebo during that period of time, and then a follow-up. There's clearly an improvement in FEV1 in the overall population, but in those groups who had a high peri-Austin level, most of the improvement seen in the overall cohort really resides in this group of 40% of the overall patients, 250 that were studied. And if one takes out the ones whose serum peri-Austin is normal, not elevated, there's lack of a response. So that led to that theory we had talked about previously, where IL-13 generated in a Th2 immune response in the lung induces the epithelium to produce peri-Austin, spills back into the serum, and the high serum level are the ones who respond to the anti-IL-13 treatment. So that is sort of in a nutshell the different immunomodulatory therapies in asthma that are on the horizon. There are many other immunopharmacologic agents, steroids, mediator antagonists, immunosuppressives besides these newer immunomodulators, so I'll just mention these, and you can look up anything about them individually because they're not worth really going through because the data is not overwhelming for any of them in asthma. Well, I should mention anti-leukotrienes are not in that category, but antihistamines in asthma are questionable. Some of these other arachidonate metabolites are also inhibitors and antagonists, are not really strong modulators of asthma. And some of them have been tested along with neurokinin, bradykinin antagonists that are borderline in terms of their importance in asthma, to be honest. Same thing with NOS inhibitors, tryptase. There have been studies, but none of them have been conclusive, and none of them have been really very exciting, to be honest. Steroids, work through the secondary inhibition of a variety of targets that we already talked about in terms of biotherapeutics as targets. And obviously they inhibit transcription by enhancing ICAP beta and acetylation of corticostones as well as activating glucocorticoid responsive elements and then having inhibitory effects on other pro-inflammatory transcriptional complexes. So steroids work at a variety of levels to get the endpoint. They also have a variety of side effects that you all know about. There are a bunch of immunosuppressants, immunomodulators that you can look at, some of which we've mentioned in the discussion, some of which we haven't, and some of which are of value in terms of treating other immune disorders, and some of which are of value in treating allergic disorders. Certainly tacrolimus and some of the immunophilin cytophilin inhibitors are critically important in the overall treatment of atopic dermatitis, that severe atopic dermatitis, and have to be thought of in that circumstance. Some of these other ones haven't really been fully tested yet, but some of them may have some value, and they're just worth mentioning here at the end. As mentioned, some of the small inhibitory RNAs that are used in modeling in vitro experiments potentially may come of value. So antisense oligonucleotides, inhibition of families of small inhibitory RNAs are things that people are thinking about. Designer inhibitors of specific tyrosine kinases like SYK, for example. SYK kinase, which is important in mast cell and basophil degranulation, have been targets for treatment utilizing useful approaches to enzyme inhibition. Adhesion blockers have been utilized, and these are surprisingly, since they're so critically important in terms of adhesion molecules to bring in eosinophils and neutrophils and basophils to allergic responses, as well as dendritic cells and T cells on a different type of time scale in terms of accumulation of inflammatory cells. It's been amazing that the adhesion blockers that have been tried over the past 10 years, a couple of which are mentioned in the Kavanaugh paper, none of them have really been useful. And I think that relates to the fact that as complex and degenerate the cytokine networks I've described in these cascades are, the chemokine receptor cascades are even more degenerate, if you can follow what I say on that, and that there's just so many other kinds of alternative pathways. If you block one chemokine or chemokine receptor, maybe you can make a model system where you're blocking the accumulation or movement of eosinophils or neutrophils. But in terms of those kinds of pathways being critical in terms of the clinical outcome, that's been a little bit more disappointing. There are many other targets that I already just mentioned that are further down the cell cycle with activation in terms of nucleotide synthesis, kinase activation, interaction amongst these different pathways that one could see. And you can see here the list of potential either biotherapeutics that block the ligands that trigger these responses, or enzyme inhibitors that potentially could be used. In spite of all this, not a lot of these things have been very, very useful. CTLA-4 Ig has been effective in RA and lupus. I don't think it's been tried in asthma yet. Any of the activation of anti-LFA1 found to be effective in MS, even though progressive multifocal leukoencephalopathy is a potential risk, especially in those individuals who've had prior viral infections. That's an experimental treatment. It's thought to be too risky for many of the asthma trials. Inhibition of a variety of these other molecules as well involved in lymphocyte and or immune cell activation have been tried. None of them have been have blown the doors off of the treatment. You see here some other ones that we've mentioned. Some of these ones that are involved in blocking the adhesions, like Omalizumab and some of the smaller molecules, especially disappointing, at least to me. There are some antibodies, MEDI528, and some other ones that are looking to block IL-9. There are a couple of antibodies that are in trial in both asthma and atopic dermatitis that will block the IL-4 receptor, so will block both IL-4 and IL-13, and some of the early data looks promising with some of those kinds of molecules made by a couple of different companies, including Amgen and Genentech Roche, Sanofi, and Regeneron. So there's a bunch of different things that are still potentially being tried. In spite of all that, I think biotherapeutics is the next wave of the real advances in treatment we're going to be looking at in the next 20 years. When I first started giving this talk, I used to say 25 years, but we haven't made that much progress in the 5 years I've been giving the talk. I think we'll need a systems biology approach to these cascades. That's a whole different other talk. But the endpoint of that is early intervention and pharmacogenetic profiling, which if we can achieve those, all this work would be worthwhile. I've always been showing this cartoon. I think we're going to be able to, eventually figure out the best way in which patients will respond best, but there's no magic bullet in any of these things, unfortunately. So if anybody has any studies that they'd like to be considered for publication, the WOW Journal is going to probably go fully open access. It's fully online at this point, open to all members of any of the member organizations of the World Allergy Organization. But we expect that we'll be on PubMed Central by the end of this year when we go fully online open access, which we're working towards achieving this year. And we also have International Scientific Conference in Hyderabad, India, which will be in December of this year. Very nice place to visit. If you read the New York Times yesterday, they had a highlight of what to do if you were going to spend 36 hours sightseeing in Hyderabad. So if you want to find out about Hyderabad, just go to yesterday's New York Times travel section, and you can get a preview. It'll be a good conference, but it also sounds like it'll be a nice place to visit for people who are interested in seeing what India is like. Okay, thanks.
Any thoughts of having that conference available online for those who maybe aren't able to travel to Hyderabad?
I think that's worth considering. I think there's some questions about it may not be online live, but certainly maybe
why not record it and post it like we do with Darkolos as a podcast. India, some organizations say, well, we won't put it online or post it because we want to encourage people to come, but most people aren't going to be able to travel to India. That's just out of the question for many people. So as an incentive, not having it available, I don't think is going to change.
Yeah, no, and I mean, we're not trying to disincentivize anybody, and we certainly think the information there is going to be very useful for people because it's an excellent scientific program, especially since I was involved in
I have no doubt
designing it. But at any rate, I think that it's it'll be a good conference, and it's an interesting part of the world. Most of the people who will attend, obviously, are going to be from the Asia-Pacific region, Pacific Rim, and even India. And there are many Indian Americans who are going to be attending as well.
I've been to Hyderabad. It's a very interesting.
Yeah. So it's got a lot of interesting things. But the conference, I think, will be worthwhile too. I am all in favor of cataloging the material from the conference and making it available. Even though I am president-elect and editor of the journal, I don't have a final say on all of that. But certainly we'll bring that up. And we're getting closer to the time where I think it'll be fully an acceptable, route.
That's one question.
Mark.
The anti-IL-1 therapies you're talking about?
Yep.
So why would someone choose anakinra over those other therapies with the issues of the daily change?
It's the only thing that's available at this point.
You can't use those other two, the other ones?
They have just become approved only for the same, but the indications for anakinra are much broader. So if you just have any patient with rheumatoid arthritis, they can get anakinra without any problem. Although, the same kind of thing in terms of limitation by insurance companies about different things, but you can make the argument that it's an approved indication, and it's a much broader group. The canakinumab and the rilonacept are only approved for the CAPS, the familial cold urticaria, Muckle-Wells, Nomans, and that's obviously a very small and limited group. To me, I think these are the best treatments for all of those other things that were listed there. Some of which we may peripherally see, like gout, which potentially comes across. There's good evidence that one of the most strongest signals to the innate immune system are urate crystals, and that's probably one of the routes in which the inflammation getting an acute gout attack occurs. So, I think all of these IL-1 pathway inhibitions are going to be important in some of these other circumstances. And for asthma, it's a natural. There's a couple reasons why it wasn't used. One was there's a much bigger experience with blocking TNF. And it was thought that there's something of an overlap with what you get with TNF with what you get with the IL-1. But the TNF receptor, the thing that's not engaged when you're blocking the TNF, is linked to a lot of the caspase activation pathways, and you probably may get inhibition of apoptosis, for example. That's probably why there's a predisposition to cancer with the TNF blockade. For IL-1, that link is much more tenuous. Well, they may be there. They both are associated with a little bit more risk for infection, obviously. But I think IL-1 is an underutilized target for atopic dermatitis and for asthma and for a lot of allergic responses. You know, in the early days, when he was doing a lot of the food challenges in the beginning with food allergy patients who had bad eczema, Hugh Sampson always used to tell me that a lot of these kids who had the bad eczema, when he challenged them with the foods that they reacted to and they had a flare, many of them got febrile, which would suggest that something like IL-1 may play a role even in that kind of a challenge. So,
Lanny, it seems to me that asthma treatment is still the most effective treatment we have is steroids.
Right.
And that's probably because they have such a global effect. They affect multiple pathways and do things sort of more at the root cause rather than at the periphery where you're dealing with individual cytokines. And yet the emphasis on these immunomodulators is very specific, this cytokine or that receptor or whatever, and that may account for part of why these are not proving to be as effective as you'd like. What about the role of combinations of these things? Wouldn't that more closely mimic the beneficial effects of steroids?
Yeah, I agree with you completely. The combination of an IL-1 inhibitor and anti-IL-13 and maybe, you know, the anti-IL-17, you start to get a more specific blockade of some of the things that you're seeing with the steroids, but you're probably going to avoid the side effects because you're not getting the steroid effects on the side effect things. You know, years ago when I first gave and thought about these areas and gave talks, to me, essentially, all these anti-cytokines are sort of building a very expensive ziggurat, if you will, to, you know, get the same effect you get with steroids. You know, it's an expensive way to get the response, but at the same time, there's no risk of, you know, cataracts or diabetes or, you know, aseptic necrosis or some of the other kinds of really horrendous side effects you get with steroids.
So are there studies going on now that look at maybe cocktails of these in the
Yeah, no. The only real combinations I think that are being looked at are the ones, some of these agents, because of the way the antibodies are made, can block a couple of cytokines. So there are agents out there that are looking at the simultaneous blockade of IL-4 and IL-13, but that's sort of concentrated in the same pathway. If you could block IL-1 and IL-17 and IL-13, that would be a combination that I think would be better. But most of the way in which drugs are approved in the U.S., even in the cancer world and in HIV, is to show individual efficacy first, and then you can show combination improvement. There's no question.
But these things might not be effective individually.
Right. Or they may only be effective in a subpopulation, and then you can build subpopulations with the different things. But, you know, it's easy to say those things, but it's, you know, costs $50 or $60 million to get the answer, and, you know, you invest that amount of money and maybe you have a 20% chance of getting a positive outcome. So, you know, that's part of how that process works, unfortunately. You know, to me, since I don't buy any stock related to any of these things, and I don't care, and I don't have any disclosures.
Your first slide said
well, no, the slide said that's why we do disclosures. But like I said, I don't buy any of those stocks, and I certainly don't have any of those ones that I listed as a potential thing. Although, obviously, I can't hide the fact that I'm an expert in this area.
Right.
Some of the products are made by some of the groups I consult with, but that doesn't influence me in a biased way because I don't really care about the companies, whatever. I want to find out a way to treat these diseases the best way. And getting to the bottom of how the disease works is the best step to get to that. And it may cost a huge amount of money. It might take a long time, but, you know, getting towards that goal is really where we want to go with this kind of thing.
How close are we to being able to use information from genomics to maybe select a cocktail that might be effective for an individual person?
You know, that's how it's been sold for the last 10 years or 12 years, maybe 15 years among the cognoscenti. But I think that's a oversell. I think if you have something like that by the year 2050, you'd be lucky. Because most of the circumstances where that works, and they're very rare, you know, a couple of cancers and, you know, some of these drug interaction things. I mean, this is like 0.03% of all the potential medical indications for genomics. So I think, you know, if you figure out the mechanisms, then you can dissect which population it works and which don't. And all due respect to those UCSF guys who did the periostin, that's all like a shot in the dark. And it worked that one time you saw it there. There are probably 99 experiments that you get the same, that's just as good an idea as a periostin, but it doesn't work. But maybe as we understand more, all those other ones that failed eventually could work. The first studies with anti-IL-5, they gave them one shot of IL-5, their blood eosinophilia went away, and then a month later their bronchial hyperresponsiveness was unchanged. And their FEV1. So this was the pulmonologist's view of the fact that these things are unimportant. But when you do the studies the right way, you actually can find where it works. And, you know, none of these things are cures for asthma. Asthma is not due to too much IL-5. Asthma is not due to too much IL-13, no matter what some people write about it. Asthma is not due to not enough TGF-beta or too much TGF-beta. Asthma is due to how these complex cascades really interact. Some of it is probably fortuitous. Some people, if they have these cascades that are activated, there may be a random event, maybe getting exposed to the right kind of rhinovirus that tips the whole thing over to the point where it's uncontrollable. And then there may be others who get something exposed and the whole thing quiets down, or maybe there's a natural response that makes all these cascades quiet down and they're not affected clinically for years. You know, I love to speculate about that stuff, but after a while I get to the point where if I keep doing that, people would probably think I'm senile or crazy or something. But
well, we all get there eventually.
Yeah, well,
obviously,
some of us get there faster than others. So I don't know.
Maybe thinking about this accelerates the process.
Yeah, maybe. Well, it makes you worry that maybe you are crazy about. But I think it's, you know, this is the nub of why we want to, you know, help these patients. You know, this is not just figuring out, you know, a jigsaw puzzle so that you have a picture on a tabletop because you put all the little tiles or pieces in the right place. The more we learn about this, the closer we are to figuring out the best way to treat patients, which is a reason to try and get new information.
Well, it appears like it's job security for clinical investigators anyway.
Well, if you can convince anybody to give you money for it, that's true.
All right. Well,
okay.
Very complex issue. Hopefully we'll get some more information and you'll come back and update us on how it's going.
And to be fair, these immunomodulatory pathways highlight the best things that you really should study in the basic immunology, both for testing purposes as well as to, you know, carry the best kind of approach to what the mechanisms of these things are.
All right. Well, thank you so much, Dr. Rosenwasser. Really appreciate it. We're going to take about a 3 or 4 minute break, and then we'll be back, and Dr. Salzman will be hopefully joining us, and he'll talk with us about bronchoprovocation tests. This is Conferences Online Allergy from Children's University Hospitals and Clinics in Kansas City, Missouri. This has been an ACAAI production. To learn more about Conferences Online Allergy or the American College of Allergy, Asthma, and Immunology, go to www.acaai.org. See you next time.